Okay, we're going to change tack a little bit now. We're going to start with our session this morning, advances in formulations. And so we have a theme here. We're going to be looking at, as we always do, formulations and processes, because of course that's very important to us all. And we have some information and people talking about excipients, because one of the problems that we face, and you all know this, is that we have a limited range of approved excipients. What do we do about that? And sometimes there's a knee-jerk response to, oh, we can't do that because it's not approved. So that's a limitation, but there are people looking at it. And so one of the first things, of course, is taking into account some of the studies that need to be done from a regulatory view. So I'm delighted that we have people who could help shed us light on this. So with that, I'll pass you to my colleague who will introduce Victoria.
Okay, thank you everybody. So the first session in our first speaker, in our session 6, Advances in Formulations, I'd like to welcome Viktoria McDonald from Albertox Consulting. Victoria's a European registered toxicologist with 20 years experience in non-clinical safety evaluation, working with major CROs and then as an independent consultant for the last 8 years. Victoria's conducted and overseen hundreds of non-clinical studies and safety assessments, novel and repurposed drugs for inhalation, as well as multiple interactions with global regulatory agencies to obtain their approval for national and international clinical trials. So I think it's fair to say that Victoria is fully qualified to educate us in this matter. I'm very welcome to hear you speak today, Victoria. Thank you.
Well, thank you. Can you hear me?
Yes.
Is it mic on? So thank you to the committee for organising a really lovely conference and inviting me here to talk to you about non-clinical safety evaluation of excipients. And you might be able to tell I've not travelled very far today. I'm a local girl. So I'm going to give a broad overview of the toxicologist's process of identifying when an excipient is considered novel and the kind of resources we use to put together a safety assessment and a data gap analysis and a testing strategy that we can use to take our new excipient into a drug product and into clinical trials. And I've got a couple of scenarios and case study examples that I'll present at the end. So excipients, these are substances that are in most, if not all, drug products. They are inactive ingredients and they form multiple functions within the drug product. So that may be to improve the bioavailability in the lungs, just to get it into the lungs. It may be to help retain it in the lungs, and it may be for other reasons like taste masking the drug. There are lots and lots of reasons why we use excipients. But unlike APIs, the overall exposure to excipients isn't very well understood because patients can be exposed to the same excipients from multiple different drug products, and even from food. They may also come into contact from these chemicals. So, for example, asthmatics and COPD patients are taking in HFA propellants from multiple products, or they are also taking in lactose or polysorbate 80 and things like this that are added into lots of different products. So it's really important for that reason that we evaluate the safety of these excipients and that they are exceptionally safe. So novel excipients, I think, Hugh did a good job of pointing out the fact that we probably need more technologies to help us deliver drugs into the lungs more efficiently. We've got biological drug products that can be quite challenging from delivery and stability. So there's a big need for novel excipients for the inhaled route. So a novel excipient is an inactive ingredient that's not previously been approved for a marketed product. And obviously that would involve examples such as new chemical entities, but there are lots of scenarios where an excipient would be considered to be novel. And that may be a modified excipient, so a polymer that is just a slightly different chain length than a precedented excipient, new co-processed excipients, or even route switch. So if you have a substance such as xylitol, which is approved for oral and topical routes, but hasn't been approved yet in a product for inhalation, then that's novel, and it needs some level of safety assessment to take it through into clinical trials and also eventual marketing. Even precedented inhaled excipients could potentially be considered novel. For example, if you use higher doses of that excipient, a good example would be mannitol. So mannitol is approved and precedented around 6 mg per day. But if you want to start using 20 mg per day, then you're going to have to provide some reassurances on the safety of that. And the same goes for approved chemicals that are in the food industry, the cosmetic industry, or over the counter. You're going to have to do some level of safety reassurances to the regulatory authorities for a pharmaceutical drug product. So not all novel excipients are equal in the amount of safety evaluation that they need. So at the top of the list is obviously the new chemical entities, which need the greatest amount of safety evaluation, the greatest amount of testing. And down the bottom of the list, you may need less testing for something that's already precedented by another route, where you can leverage safety data from literature, or from other safety databases, or regulatory approval and other indications or routes. So it's a spectrum. So when we come to determine whether something has any extent of novelty to it as an excipient, there are several resources that you would first check from a toxicology review. You would obviously look at the Handbook of Pharmaceutical Excipients as a really good resource. So it has a lot of information on the kind of routes, the routes that have been used for the excipient. There's some basic safety information usually. It'll tell you the geographical locations where there may be approvals already. And then there's also the FDA's inactive ingredients database, which is a great resource to find out whether your excipient that you're about to evaluate has any precedence by any route and what those maximum doses might be in approved products. So you essentially can just type in the name of your excipient and it will bring up the routes, the dosage forms, and then the maximum potencies. And if you're very lucky, it will tell you the maximum daily exposures, but not always. And sometimes you have to go digging into drug labels and looking for what doses are already approved for this excipient. And actually, the FDA have drafted up guidance on how to use the inactive ingredients database, because many applicants refer to this database to show that there's some precedence for the use of an excipient. And I think they've obviously felt the need for the guidance because some people may not have been using it appropriately. And it gives quite a lot of pointers on the fact that you would still need to support certain excipients with information. So it's a useful resource. So in terms of regulatory requirements for excipients or novel excipients, the ICH safety guidelines, which drive a lot of what we need to do non-clinically for APIs or drug products, don't really specifically go into novel excipients and what to do about those. The EMA don't really go into detail either. There are some general advice that you should be qualifying your excipients and making sure they are safe and conducting the relevant studies. But there isn't really an awful lot of detail. But the FDA, they have very specific guidance that is holding novel excipients up to the same level of scrutiny as an API and demanding the same amount of safety data as you would for your drug product and for an API. And then I found this really good safety guide, which is useful. It's from the International Pharmaceutical Excipients Council. They published this in 2021. And in fact, that's quite a really good guide on the sort of information that you would require to have on your excipient. So safety evaluation, what do we mean by safety? Well, that's to say that a substance has an absence of undesirable or harmful effects. So an undesirable effect may be that the substance causes an increase in heart rate or a change in respiratory function, or a harmful effect that is actually pathological and it's causing damage to the tissues. So we need to find all the data, put all the data together that can give us the reassurances on this to establish what is safe and how much is safe. So how do we identify existing safety data? Well, we must make all efforts to try and identify it before we start commissioning animal studies. obviously from an ethical perspective, that's the right thing to do. We need to know what the dose, we need a handle on the doses of the excipient, because sometimes I've seen examples where use of a colorant, for example, has been added to help blind a vehicle, a placebo in a clinical trial, but the levels used were at at levels that were below thresholds of toxicological concern, and therefore didn't necessitate any specific testing. And obviously there's literature searching, PubMed and all the rest of it for peer-reviewed literature, but there are really good safety databases that hold a lot of toxicological information. So the European Chemicals Agency, since the introduction of REACH, there has been the need to have registration dossiers for certain chemicals, and obviously many excipients are also used in other industries and may have already been tested for inhalation toxicology due to occupational risks. So you may find that you can locate some information in these databases, and also from cosmetics, Cosmetic Ingredient Review. They put together a lot of safety information, and sometimes that will also contain inhalation toxicology information. And the OECD as well, they've done a lot of reviews of chemicals that you might find something in there. And when we're looking for this information, we're really trying to gather specific data sets. We don't want to start developing an excipient that is genotoxic. So we're looking to make sure that, that there's some testing being performed or not. And then the metabolism and excretion, does our excipient potentially inhibit the metabolism of other drugs? Safety pharmacology, so we want to know whether our chemical can change cardiovascular functions, respiratory function, and then obviously anything repeat dose toxicity, even if it's from the oral route, still useful because it tells you about the systemic toxicity and the potential target organs of interest for that excipient. And if there's any reproductive toxicity information out there on this chemical, because we don't want to start developing a teratogenic excipient into our new drug product. And obviously carcinogenicity, irritation, sensitization, immunotox, and all these things would go into this assessment. And it's possible in certain scenarios that abbreviated safety data is acceptable, where we can't always fill information into all those data sets. And that may be such as life-saving therapies, large polymers where we could use a read-across approach from a precedented polymer. Existing human use, you could leverage a lot of that data, and GRAS status, which is when a compound is generally recognized as safe. And generic products, although generic products, they don't often put novel excipients into the product, but what they often do is they put a new excipient or an existing excipient with an existing API, but those two haven't been in a product before. So now we have a situation where we don't know if the pharmacokinetics and the efficacy will be the same. So there may be some liabilities to look at clinical safety testing. So when we've done all our literature searching, looking at all our tox databases, and we gather up all the data into our data sets, we can look to see where there are gaps in those sets, and we can develop a strategy for testing that would meet regulatory requirements. So sometimes that strategy could be literature only. It may be that it's literature plus a little bit of clinical data. It may be literature with specific safety testing, or we may have to go into a full testing regime depending on the novelty or the extent of novelty of that excipient. So I've just got a couple of scenarios here where we may want to consider that xylitol is not currently approved in any inhaled drug product, although it has been tested in lots of ways. Xylitol would be considered novel for the inhaled route. And imagine we're going to take this into an IND in the US. What we would do, we would leverage the GRAS status, because xylitol is used in food, so it's generally recognized as safe. And that's to say it's not genotoxic, it's not reprotoxic, and it's already precedented in oral, sublingual, and topical drug products. So we can leverage that, and we've got a lot of information already And in fact, for xylitol, this literature searches will reveal there's 14-day inhalation tox studies, there's 28-day rat studies, and that's enough to support a phase 1 study. There's also evidence of medical use, of sort of and healthy volunteer studies in Europe. So we can leverage all that information, and it may be that all we now need to do is our drug product toxicology, just as we would with any other product, but with some extra additions to our study designs, such as ensuring that when we run a toxicology study, that we've controlled for the presence of xylitol by adding in air controls. That means we maybe have a 5-group toxicology study as opposed to a 4-group tox study. And we may have specific concerns for xylitol would be, could it cause bronchoconstriction? So we may want to add in additional respiratory function checks for xylitol. And when we come to submit the IND, we don't just submit our toxicology studies, we would demonstrate that we have looked into the genotoxicity, we've looked into all the background data on xylitol and all the precedented status, and we would put that into our IND as well. In another scenario, in a new chemical entity situation, which is what mankind did when they developed technospheres, the fumarole diketopiprazine, which was a new chemical entity for inhalation to enable the delivery of insulin into the lungs for rapid uptake. And quite interesting, because insulin is quite precedented in terms of we know a lot about its pharmacology, we know what the main risks are in terms of exaggerated pharmacology, and the excipient is the novel part here of the whole product, and mankind had to spend a lot of time and a lot of effort to demonstrate the safety of the FDKP. But in this scenario with a new chemical entity, you would want to start with, if you had an idea that we want to put this new chemical into a drug product, you would obviously want to know about whether it could be mutagenic as a starting point. And you could use in silico checks such as QSAR, which is qualitative structural alert reactivity, and that will tell you if there's any parts of the structure that could potentially disrupt genetic material and cause mutations. So if that's looking good, then we can go into in vitro gene tox testing and make sure it's not genotoxic in standard bacterial mutagenicity assays. And then we can take it into other assays and go in a stepwise process to make sure we're not betting on the wrong horse, and we've got no problems with the metabolism, that we're not going to start inducing and inhibiting the metabolism of other drugs. And also cardiotoxicity, HER gases, it's not going to cause sudden death of somebody or sudden cardiac arrest. So there are a lot of screening tools that you can use early on before you start getting further and further into development and start to use, get into your animal toxicology studies. And in fact, that's what mankind did. They ran a lot of assays where they had the Afrezza drug product, but they also ran alongside was the technospheres on their own. And they ran all the toxicology with drug product, and they would have had to ensure that they had some level of controls in there for the FDKP. And another point, they would have had to ensure that the amounts of FDKP that would be given to patients were covered at a dose multiple higher in the animal toxicology studies. And they would also, because there would be no existing monograph for this compound, they would have had to get into all the usual CMC characterization of impurity profiles and all those things. So they had to do an awful lot of work to demonstrate the safety of this novel excipient in Afrezza. So just to summarize, we know we really need novel excipients. They're needed for the innovative inhaled drug products that are being developed today. But the extent of the safety evaluation is very much dependent on the extent of novelty of that excipient. So it's along a spectrum, and you need to harness all the other tools and all the other information that's out there to be able to put a proper strategy together. And I think in these situations, and depending on the extent of novelty of an excipient that you might be developing in your product, it's always good to get regulatory and scientific advice before making any major submissions. And with that, I'd like to thank you, and I hope you've had a lovely time in Edinburgh, and take some questions.
Thank you, Victoria. Great presentation. And I love that curve you put up between the sort of the amount of safety data needed and the degree of newness, and not assuming you need everything, but to do the gap analysis to decide where you are on that curve. So that was great. So folks, any questions from the audience? I know it's nearing coffee time, but there must be something there that you wanted to ask.
Yeah, that was really a nice talk. So is there a compendium? You showed a handbook of excipients. Is there a compendium of inhaled excipient doses, what products they've been used in, that we all can go to?
Not that I'm aware of a specific inhalation compendium, but just these excipient handbooks and the use of the inactive ingredients database. And I think maybe some companies do keep their own records and they've done their own digging, but I'm not aware of any specific listing.
That would be really helpful.
It would.
Thanks.
Who's got a question? You
Hi,
ask.
back there, please.
Was that the same question?
No.
Eddie? Okay.
Hello, Eddie French. I just wanted to thank you for doing that, because to point out that there are routes for excipients that are not sitting in the I2D list is very important, because I've seen it as probably one of the biggest handcuffs to innovation in the formulation area for many years, and companies' obsession with if it's not on that list, you do not use it.
Yep.
So the fact that you have pointed out routes where things, sometimes with little effort, sometimes with huge amount of effort, can be taken through, could open up a lot of areas. It's kind of like having a great drug and then forced to use a 50-year-old formulation because it's the precedented thing and it's the most frustrating thing going. So just to keep that in mind in promoting what you do, that there are ways forward to do things for companies, I think it's very important. It's more of a comment than a question, I'm afraid.
Oh, thank you. No, thank you. No, I think it is important because I think you're right. And it's also, we have to avoid doing unnecessary testing where possible as well. And there's a big drive for 3 Rs, which is obviously the reduction of use of animals. And there's also new technologies that can be employed in vitro toxicity assays for specific to the respiratory tract. There's 3D tissue models that you can use to replace animals in certain circumstances, and they can give you early data and indication of whether your substance is going to be, safe or not.
Thank you, Eddie. I think there's one question down the front here.
Hi, that's a very nice talk, Carsten, Trinity College Dublin. For this simple academic, can you give me an estimate of the 2 case studies that you showed, what this roughly would cost?
Oh,
Yeah,
no.
just because, okay, so let's do that.
Oh, well,
5 million, 50 million.
Well, to run a non-clinical inhalation toxicology program in full is very expensive. And it's more expensive than running oral and IV because there's a lot of wastage of the testing, and there's a lot of technical expertise that needed to generate aerosols and be able to get them into the animals and quantify the doses and all the analytics that go along with it. But it's difficult to give an estimate, but for the costs are going up all the time because demand's really high. But it's millions. I mean, it can run into, and it depends on the duration of your treatment. Are you developing for a chronic indication or an acute indication? So a chronic indication you would have to go all the way through to carcinogenicity studies where you're running animal studies for up to 2 years, which can get extremely expensive. So we're talking millions for,
Okay, any more questions? The one at the back, please.
Hi, Paul Hardman from Broughton, and actually this follows on from the previous question. If we had maybe 2 or more novel excipients, would we be able to run the in vitro assays, the in vivo studies in combination, or would they all have to be separate?
The in vitro, well, you develop your own strategy and you can say, well, I want to do things in a stepwise process, so I'm going to run an in vitro assay. You may want to screen 2 or 3 novel excipients and then say, well, I'm going to pick the one with the least in vitro toxicity. You can also look at permeability of your, if you want to look at how your drug would be absorbed through cells, you can do that too in vitro, and that might help guide you towards picking out your final novel excipient before you go into those in vivo studies. And sometimes, depending on the extent of the novelty, you may be able to say, well, we're going to run an in vivo study and we're going to complement that with an in vitro assay, but it all depends on how much data is there already in the public domain, and how much you can justify that. Because the expectations are always fairly standard, and you've got to challenge that, but you've got to have the data there and the strategy and the planning and the scientific justification to do that.
Thank you.
Great, thank you.
I have one question, and I'm going to apologise if it is a bit naive, but I'm curious. Obviously, we're in a landscape of emerging knowledge, and as we find out new information every day with better analytical techniques, etc. And what impact do things like that have on the ability to repurpose established toxicity data? I guess the example I'm thinking of is things like nitrosamines, which is a fairly curveball in the industry in terms of our understanding and safety. And is that something that you really need to make sure you factor into any gap analysis you do, or any thoughts that you have on things like that?
Sorry, I wasn't clear.
I think nitrosamines is...
As an impurity, or
the potential interactions and safety implications of that, and what does it mean in terms of the ability to repurpose things like tox data from established excipients?
I mean, it's difficult to say in an example like that, but you can obviously, all of these things come down to the total exposure. So as long as you can get a handle on how much nitrosamines you're going to be exposed to and whether that's considered safe or not, and you can assess that from literature, you can use toxicological thresholds of concern, you can factor in a risk-benefit and all those things.
Okay, thank you.
Good. Thank you. So thank you again, Victoria. Okay, guys, it's now coffee time. We had a great morning. I just want to thank the speakers again. We had Magnus, who has told us about how to successfully foster innovation. Hugh talking about a range of technology developments, and Victoria, of course, teaching us more about these sort of preclinical considerations for new excipients. So we will continue, I think, at 10:35 with 11:35. There's an important point. Okay, thank you.
Thank you.